Hello and welcome to today's webinar, Easy, Efficient, and Scalable TEM and SEM Sample Preparation with the mPrep system. My name is Stephen Goodman. I'm the Chief Scientific Officer of Microscopy Innovations. In the original broadcast, viewers were able to ask questions. However, for this recorded webinar, you will of course not be able to ask questions. However, we would be happy to hear from you with your questions or comments after the webinar or at any time. The agenda for this webinar is to introduce the mPrep system and show it to some degree in comparison to existing methods of sample preparation. The next part will be to look at efficiency and reproducibility, what the mPrep system provides. Then I will explore some application examples in TEM and SEM, followed by how to manage complicated preparation protocols for TEM, SEM, and some other imaging modes, followed by summary and conclusions. Microscopy citations have increased every decade. As we can see here, there was about a doubling between 1960 and 1970, and already in the 2010s, we have achieved a number of citations where the word microscope or microscopy was used in PubMed, National Library of Medicine. You can see that this level is pretty comparable to all of the 2000s. So why? This is because microscopes have gotten better and more labs are using them to address more questions. But microscopy sample prep has barely changed over this time. The process is still intensely manual. TEM sample prep uses manual fluid exchanges using manual pipetters and commodity vials like scintillation vials or microcentrifuge tubes. Even automated processors require a lot of manual handling as tissue is placed into and out of baskets and into embedding molds of different types. The timing for reactions are quite inconsistent. It's very difficult to generate exactly the same reagent time when one is processing multiple specimens in a scintillation vial or another commodity vessel. Block handling requires a lot of work, and orienting specimens using flat molds is approximate at best. Reagent consumption can be excessive since it is determined by the container and not by the needs of the specimen. Grid handling, as we all know, is extremely tedious, prone to error. It's easy to drop a grid, mix up a grid, and such. And throughout the process, every documentation, every process change has to be documented as one move grids from a grid box onto a droplet. We have to note where this is. And of course, since we have all of these myriad vessels, it can be very, very difficult to track your specimens or your grids as they're moved into and out of different containers. The mPrep system is different. It's based on two capsule types that are specifically designed for the needs of microscopists. These vessels are not co-opted from another purpose. The specimen capsule, shown here diagrammatically, and the grid capsule, which will be shown in a moment, are purpose-built. Let me show you how these work. So a specimen is simply dropped into or placed into the mPrep S capsule, microprep for specimen capsule. The capsule has 300 micron holes on the bottom. Once the specimen is within the capsule, a removable adjustable screen that also has 300 micron pores is placed into the capsule to entrap the specimen. Once specimens are in the capsule, they can be barcode labeled, as we see here, with a 2D barcode or an alphanumeric label. And then for fluid processing, the specimens are specimen capsules are attached to micropipettes, ordinary pipetters like a pipette man, and the fluids can be drawn in and out. The mPrep G capsule is an entirely different design, but operates in very much the same way. There are 2 grid slots. Grids are inserted into the capsule at the microtome. And once they're inserted, the capsule can be barcode or alphanumerically coded, labeled, and a micropipette is connected to also deliver the fluid reagents into and out of the capsule to treat the reagents. After fluid processing, the capsule can be closed to safely encapsulate the grids within the mPrep G capsule. As I said, this is a purpose-built labware that's designed for the needs of microscopists. The capsules can be always labeled, therefore the specimen is or the grid is always labeled. By using pipettes, we can deliver reagent precisely and conveniently. And there are a whole suite of biotech tools and instruments that we can now utilize in the microscopy lab. This is what the capsules look like. Here we have a specimen entrapped into the mPrep S capsule here. Here we can see the 300 micron holes in the bottom, 300 micron holes in the screen, and here is a screen positioned within a capsule. Here's a specimen that's embedded in epoxy within the capsule, and here is a capsule that's attached to a single channel pipetter for fluid processing. The specimen is viewable here. Here we see the mPrep G capsule. There are 2 grids and 2 separate slots. They're labeled A and B, as we can see A and B here. These capsules attach directly to the pipetter. Here we have a grid being stained with urinal acetate. The grid looks a bit oval here because the plastic acts a little bit like a lens, so it distorts what the grid looks like. And here we see the labels on these. Let me further explain how these are used with respect to an EM specimen and TEM grid workflow. So, in a biology study, the specimen would be placed into the mPrep S capsule, entrapped with a removable adjustable screen. The capsule is then connected to a pipetter, like a pipette man. Once it's connected, all the fluids for the processing, the fixation, dehydration, and embedding resins are all exchanged, as shown here. This pipetting process is extremely easy to do, so we can also engender agitation during these processes by simply doing a full or a partial volume fluid exchange to do agitation. Once the specimen is in resin, the capsule is ejected, placed into a tightly sealing container so it seals the bottom of the capsules, and then this assembly is transferred to the oven for curing the resin. Once the resin is cured, the specimen within the capsule is directly clamped into the microtome chuck, and the block is faced through the capsule, and the sections are obtained. So this is where the mPrep S capsules are used. Of course, we can also do SEM sample preparation, and we would be taking this through a fluid processing and not through embedding. So once sections are obtained, they're placed on TEM grids in the usual manner. The grids are then inserted into the mPrep G capsule. The capsules are then stored into the mPrep capsule grid box. And here we can see the capsules are labeled, diagrammatically, of course, and the box is also labeled. And here we have capsules that are are closed, and here we have capsules that are open, and you see the grids in there. The grid slots will retain the grids even when the capsules are open, and I'll show that a little bit more later. When the time comes to stain the grids or immunolabel or some other fluid processing, the grids in the capsules are simply connected to the pipetter, and the fluids are exchanged to do the appropriate staining. So this is where the mPrep G capsules are used. So let's explore a little bit more about the mPrep S specimen capsules. This is the screen. Here's the tool that's used to put the screen in and out. And obviously here is the capsule. The screen has tabs, and there are fingers on the insertion tool that grip the tabs in a very simple manner, like a bayonet fitting, to allow the screen to be positioned within the capsule. The screen can be positioned all the way to the bottom of the capsule, in fact, even touching to entrap a very small sample at the bottom. Here we show it about 2 millimeters from the bottom, and the screen can even be positioned all the way to the top over here with nearly a centimeter of distance if one has a very long specimen and wants to do something a little unusual. The screen has a concave side and a convex side. Here we see the convex side down for entrapping. The screen can also be inserted in the upside down position to put the concave side over to entrap a specimen where that's more appropriate. I'll also in a moment show how the screens engender orientation. So how they're used. So diagrammatically, here's a specimen that's entrapped into the capsule. We then, once the specimen is loaded, we can attach an mPrep F filter coupler. There's a filter here like a pipette tip filter, and that prevents accidentally drawing things like embedding reagents directly into the pipetter. Gives you a little bit of headspace above the fluids as well. So the sweet spot for the volume for the use of the mPrep S, it can be as little as 10 microliters if your sample is sufficiently small, to a maximum of about 150 microliters. So let's now explore the orientation using the mPrep S capsules. Orientation is most easily done, and in some cases only done, using the mPrep workstation. So the workstation is a large flat substrate of soft polyethylene that's designed for dissection. It's all one focal plane, so that you can place this under a dissection scope and be in focus at anywhere on the table here. So there's a location here for capsule and screen storage as you're working. There are these dissection wells, which you can fill with water, buffer, or glutaraldehyde fixative as you're cutting and preparing your samples. You can also drip fluid on where the specimens are inserted, and that occurs here. This is the insertion tool, and here we see a screen in the insertion tool there as well. And this lever here actuates a function in the screen that allows one to orient samples. Let me show you how that works. So we'll zoom in here, and we see there is the insertion tool right here. There's the screen, and here's the workstation around this area here. When we press down on this lever, the screen opens up a slit, and a long, thin sample, like a piece of biological tissue, muscle, skin, nerve, plant parts of plants, and many, many others, fibers, can be inserted into the screen, and then by releasing the lever that's in the front of the workstation, the screen will now clamp down and hold the back end of the sample. So now the sample is held vertically within the screen, and then to load the capsule, here's a capsule. The capsule is simply dropped over the insertion tool, rotated slightly counterclockwise, and lifted off. And we see the specimen is now loaded into the capsule, being pinched in by the screen. After the samples are loaded, they are placed over on the sides over here in these wells that can retain fluid, either on this side or on this side here. But let's look at the thinnest possible sample that can be easily clamped. Here we have a human hair that mics to about 40 microns thin. So we can orient quite thin samples. Now, because this is a very, very skinny sample, if it were not pinch mounted, potentially it could fall through the 300 micron holes. But if it's pinch mounted, that's not going to happen. And we can utilize this feature to hold other kinds of samples as well. For example, a small drop of agar could be placed on the screen here to hold that sample and keep it from moving out of the capsule. So even smaller samples can be handled. And as I mentioned here, there are wells on the side to keep specimens wet. The spacing between the wells is the same spacing on multi-channel pipetters to allow very easy loading of the capsules onto pipettes. And I'll show you how we do fluid processing, which is done with multi-channel pipettes. So here we have a 12-channel pipetter with 12 mPrep S capsules filled with urinal acetate for en bloc staining, and this is above a reagent reservoir. 100 microliters of this en bloc stain was pipetted into the capsule, and it's held in there just like your child will hold fluid in a straw when you're out at a restaurant to embarrass you. So a little drop might form on the bottom, but as you can see, the specimen is fully immersed in the stain. So one can use reagent reservoirs like these for fluid processing. One can use ordinary 96 well plates. One can use, in this case, a 12 well plate. And during the reactions, it's very, very easy to use an ordinary lab stand to pipette up the fluid into the capsules, and then just hold the pipetter in the reservoir for the desired reaction time. And at any time, one can press the button here on the top to cause some mixing of the fluid to create agitation. And so one may begin the process in one row, pipette up the fixative, pipette up a rinse, pipette up the secondary fixative, etc., all the way through. And it makes it very easy to track the process, and takes very, very little time. And at some point, one may want to take the capsules and transfer them into an oven or some other device when they're fluid filled. And as I showed in an introductory slide, one simply ejects the capsule into a mold that seals the very bottom of the capsules. And this is a product called the mPrep bench, which is a silicone 96 well plate. And when it's filled with capsules, it works just like an ordinary 96 well plate. And so fluid-filled capsules can be transferred into an incubator. For example, if one is doing a very long-term incubation of live cells or en bloc antibody labeling, can be transferred into the EM microwave for EM sample acceleration, processing acceleration, or into an oven or a vacuum oven to cure resin. We can also transfer capsules into a critical point drying apparatus using the mPrep CPD holder. So this is a block of metal. The capsules are inserted into the CPD holder, and the fluid can flow all the way through the bottom of this for very efficient exchanges of transition fluids. And this holder has a cover, which you don't see here. I'll show that later. But this holder can also be used for certain cryogenic protocols. So here we see mPrep S capsules inserted in the holder for critical point drying, and here we have mPrep G capsules with grids being processed, in this case in a cryogenic protocol, but this could also be done through the critical point holder, through the critical point dryer. So let me talk a little bit more about the mPrep G capsules. There are two slots here. It's a little hard to visualize them. As you can see, the slots just grip the very edges of the grid so that the entire grid surface is exposed to stains or labels. The grid tight slots hold the grid securely. Effectively, they friction wedge and spring wedge the grids into place. And this capsule can be turned upside down and dropped on the floor, and these grids would still stay in. So we can place one or two grids in labeled slots, labels A and B. It takes 35 microliters to stain the one or two grids in the capsule. The capsule is effectively sealed. It's sealed at the top by the pipette. There's a 0.5 millimeter hole at the bottom. If we simply place the capsule on top of a piece of parafilm or in the mPrep bench silicone holder, the bottom is now sealed, and because it's sealed staining, this reduces contamination and precipitation. We can close the cap to hold the grids in place for archival storage, and of course the caps can be labeled, and I showed you the capsule grid box as well. So here we have one or two grids being stained on a single channel pipetter. The mPrep F filter coupler is used. Here's the filter here, and again that prevents you from accidentally drawing reagents into the pipette mechanism. The grid capsules can be stacked up into multiples. Here there are 4 of them stacked up, drawing from a single reagent reservoir, and 8 grids are being stained on a single channel pipette. You can also place the grid capsules onto a multi-channel pipette, and here 8 or 16 grids are being stained with an 8-channel pipetter. If you stack the grids on multi-channel pipette, Using a 12-channel pipetter, you could actually stain 96 grids at once, which would be a rather large number of grids. Now let's go back to the mPrep S and let's discuss reagent efficiency. So this is the way I was taught. Take a sample, place it in a scintillation vial or another suitable commodity container, and make sure you have enough reagent to cover the sample, which in a scintillation vial requires about 2 mils. Well, I was also taught that the specimen should be smaller than about 1 millimeter in every dimensions, and so that means the specimen volume is about 1 microliter. And I was taught, and I believe this, and this is still taught in textbooks like Bozzella and Russell, the volume that's used for processing a sample should be 10 times the specimen volume. So for a 1 microliter volume specimen, that means only 10 microliters should be required, yet routinely people are processing in 2 mils. Well, here we have the mPrep S capsules attached to the pipetter with 100 microliters. So we're at 100 times the volume of the specimen. So this should be more than sufficient for reaction, and indeed it is. And so we can reduce this consumption from 2 mils per specimen down to 100 microliters per specimen. And in fact, we could even reduce this further. But typically, people use about 100 microliters for fixation and solvent reagents, and usually about 200 microliters for resin in order to have a sufficiently large block, and because, of course, it's very difficult to handle small volumes of viscous resins. If we process 8 specimens here, using this fluid volume, it takes 30 mils to process 8 specimens. If we use scintillation vials and this kind of cover, it would require over 600 mils to process those 8 specimens. And if we use microcentrifuge tubes with 1 mil per specimen, it's about 300 mils. So we can reduce reagent volume compared to microcentrifuge tube processing from around 300 mils down to about 30 mils quite easily, having a 100-fold excess of volume of reagent, which is more than sufficient for reaction. Well, not surprising, things work just fine this way. And in fact, because we're not changing really the chemistry, we're just changing how the chemicals are delivered, we get results that are comparable to the best of any other methods. So here's an example of kidney prepared entirely and stained entirely using mPrep processing capsules. So a light micrograph TEM image. Let's take a look at the efficiency in terms of this process. So here was the protocol that was used from Karnofsky fixative through an Ippon spurs resin. The specimens were not oriented in this particular case. They were entrapped in the capsules held near the bottom. 100 microliters were used for the processing. Actually, this is a 12-channel and an 8-channel was used for this particular study. Processing was done with reagent reservoirs like this and like this. And the specimens were embedded in resin using the mPrep bench, and we see part of it here with 8 specimens sitting in there. Because the specimens are directly chucked in the capsule into the microtome, there was no need to cut them out of a beam capsule or some other device, and that, of course, saved a little bit of time. Here we can see that the capsule's been cut away, and here we see the block faced and ready for sectioning. So let's look at the overall efficiency and compare the processes. So with mPrep S processing, the specimen was always labeled at all times, so the tracking was very easy. Because we used a multi-channel pipetting for the processing, every specimen saw the reagents at exactly the same time, so they were handled all together and the timing was identical. The number of user operations, the times that a technician was picking up a pipette in order to make a reagent exchange in and out of an individual vial in the case of vial processing, or all the specimens at once using mPrep processing. As you can see, the number of user operations dropped by nearly an order of magnitude. The touch time, the time that the technician is actually handling the samples, drops by several hours, and the reagent per specimen, as we discussed, drops about by an order of magnitude. We can also look at the processing efficiency for TEM grid staining here. So here we have a grid being prepared at the microtome. That grid is then inserted into the capsule at the microtome. Once it's inserted into the capsule, it's stored into the grid box. And as you can see, everything is labeled. Every individual capsule is labeled with a barcode and an alphanumeric code, which is wrapped around the capsule and not visible here. And the box itself is labeled with, so one knows where to put that capsule into the box. And then, of course, the box is labeled by the, in this case, by the kind of sample in the study. Processing of the grids were done all at once on a multi-channel pipetter using reagent delivered from a reagent reservoir. It could also have been done by stacking, as we see here. And then the grids are removed, and the only time that they have now been touched since they were placed into the capsule is when we go to take them out of the capsule, as we see here, and place them onto the TEM stage. So we can look at the staining protocol here, and we can count up the total operations. As we see, the total operations, if we were doing droplet staining, is something on the order of 160 operations compared to the mPrep operations of about 55. But that doesn't even tell the full story. So every time each of the 16 grids are prepared, they have to be picked up and moved one by one by one. Whereas with the mPrep processing, most of the operations are done in tandem with one motion of the pipette. Because the grids are safely encapsulated and held within the capsules, the amount of attention that is required to move a grid from one stain to the other is pretty minimal compared to the amount of attention that's required to pick up a grid with forceps and put it from one droplet to another one. And of course, all the staining times are identical with the mPrep processing, which of course they are not with droplet staining. We also cut out one step. We don't need to use potassium or sodium hydroxide because the capsules are sealed. A step that's optional with the mPrep processing is blot drying. In the case of mPrep processing, blot drying can be done simply by inserting a piece of filter paper into the capsule and touching it to the edge of the grid, which doesn't require touching the grid. But it's not necessary. One can simply air dry them. And as we pointed out before, the results are as good as any. Here's a result from the University of Toulouse from Dr. Ben Maradi there. Here's a result from the electron microscopy training program at Madison College, where technicians learn the art of EM. Here's an example from the Wisconsin Veterinary Diagnostic Laboratory of vampire bat brain. And here's another example from the School of Veterinary Medicine at the University of Wisconsin here in Madison. And finally, here's an example of negative staining that was done within the capsules. This is a rather different study. Form-var grids were inserted into the capsules. They were then treated with a reagent to improve the adhesion of DNA. The DNA was then absorbed into the capsule. The DNA was then negatively stained in the capsule, and the grids were removed prior to imaging, and they were rotary shadowed prior to imaging. And of course, that did not occur in the capsule. So again, during this webinar, questions can be asked, and a question came in via text chat. Asked about stains, how to handle stains like lead, and is there a problem with precipitation? In general, no, there is no problem with precipitate, but even like with droplet staining, you need to use the best quality stains. They need to be freshly prepared, freshly filtered or centrifuged, whatever is your protocol, and they should be placed in a reservoir or a 96 well plate to pipette them up immediately before use, or they can be placed in such a vessel and covered before use with a sheet of parafilm or some other kind of lab film. So let's now look at a couple of applications for SEM sample prep, one with tissue and one technique, also tissue with cryoplaning. So here we have an advantage that I'm going to show of SEM processing using the mPrep capsules. So a specimen can be pinch mounted and oriented in the capsule using the screen in the workstation. And once it's in there, all the fluid exchanges now occur with that sample suspended in the middle of the capsule for fixation, dehydration, critical point drying. And so here the capsules, either the specimen capsules or the grid capsules can be done, but we're talking specimens here. When they're placed in the capsule, this lid make sure the capsule stays into the holder. And then after the critical point drying, one simply uses the insertion tool or the workstation to remove the capsule, and then using forceps, place the screen holding the sample onto the stub, where the screen is now coated with a conductive paste, carbon paste, or similar, again, whatever you prefer. And that's essential because the screen is polypropylene and would charge. So this provides a good ground between the specimen and the stub. So in this kind of a sample preparation, the specimen was only touched once when it was inserted into the capsule. From then on in, the specimen was entirely suspended safely within the capsule, and all it saw were fluid exchanges. And of course, with SEM, we're looking at the surface, so not having a damaged surface is certainly advantageous. So let's look at a particular example of a tissue that was prepared in this way. Samples were mounted, processed in an 8-channel pipette. 6 specimens were done because that's the capacity of the mPrep S holder that fits into the TEM critical point dryer, as we see it sitting in here, ready for processing. And here's one of those samples that's been mounted. So here we have a tissue sample that's pinched in the screen. And here we see the screen, and we can see that some of the holes that are used for the fluid reagent delivery have now been covered up by the conductive paste, and some of them have been made smaller by it. So we can zoom in on this end of the tissue, and we can look at the tissue in this case. And in this particular example, if you're interested, a conformal wound dressing that incorporates silver nanoparticles for antimicrobial action has been applied to wounded skin. And really the question to be asked, the research question, is how conformal is this coating? And here we see the coating, and here we see the tissue, and we see it's very, very difficult to even see the interface between them, which occurs right about here between the wounded skin and the coating. So let's look at another example. This came from Bill Plumley, who's now at Ohio State University and was published last year in the Proceedings of Microscopy and Microanalysis. So what Bill did was he took the mPrep S capsule and he modified it. He cut off the very end of the capsule. He then used the workstation to hold a screen in the capsule and then mounted a tissue sample with the screen and then slid the capsule down over it so that the ends of the, so the end of the capsule was now below where the tissue is. So the tissue is now standing proud of the capsule. And here's a photograph of what this looked like. So here we have a tissue sample, and this is a woody plant tissue, so that's why it looks the way it does. And this was kept wet until processing. And the sample was rapidly frozen in a cryogen, in this case liquid nitrogen slush. And because the tissue extends proud of the capsule, the chilling rate of the tissue as it hits the cryogen is very, very good. So once multiple samples were prepared and then stored in the cryogen, liquid nitrogen, at this point, they could then be transferred to the cryomicrotome. Directly, the capsules were then directly clamped into the microtome chuck. And the front end of the specimen was now faced through to expose an internal surface. After multiple capsules holding multiple specimens were cryosectioned, they were then all freeze-dried. And then the screens were used to transfer the specimen directly to the SEM stub, and this is what it looks like. So they were transferred and then conductively mounted and sputter coated. So here we see the tissue samples being exposed. And this is the kind of micrographs that one can obtain. Here's that plant tissue. It was Christmas cactus, and we have a cross-section through it at low and high magnification. And here we have canine kidney that was prepared in the same way. I should mention the kidney was prefixed prior to this process, glutaraldehyde osmium fixation, and then extensively water rinsed prior to the cryopreparation. So there were no questions here, so I will move on. And now let's take a look at some complicated protocols. So what do I mean by complicated protocols? Protocols that have a lot of different steps and require very great attention in order to track them, and where one has many specimens that are prepared in different ways. So an advantage with the mPrep system is that we can use a template for the processing. So we can use biotechnology labware tools for microscopy. Well, the major tool in this case are the multi-channel pipetter and a 96-well plate. So this paper template looks like a 96-well plate, and one simply downloads this template and fills out the reagents that are going to be samples are going to be treated in order, in this case for an immunoprotocol. And then a 96-well plate is placed over the template and then filled with the appropriate reagents. And this setup would be for processing 12 specimens at once, or 12 grids at once, stepping from the first reagent to the next reagent to the next reagent. But we can also rotate this template 90 degrees and process 8 at once. And then during these reagent exchanges, the lab stand makes a very, very convenient way to hold the capsules in the 96-well plate vertically. So let's take a look at a typical immunolabeling protocol here. So top to bottom, we have this immunolabeling protocol. We can load mPrep G capsules directly from the mPrep grid box onto a multi-channel pipette. So we have all the capsules fully loaded, and then one lays out our reagents. So we can lay out our cheap reagents like phosphate buffered saline and reagent reservoirs, or water for that matter. And then we lay out our expensive reagents in a 96-well plate. And this is laid out for orienting, in the orientation for processing 8 capsules at once here. So this is color coding, matches this color coding over here. So the first reagent step here after the PBS is the glycine PBS, then a blocking reagent, etc. And we can step through this here. And then the next part I want to show here. So we have the sequence from top to bottom, and then we have any titrations or any samples that we wish to prepare differently laid out in this axis here. So we can have a high concentration of secondary antibody, a low concentration of antibody, and then maybe no antibody or some other controls over here. And one simply pipettes up from row to row to row with, of course, the appropriate timing and any appropriate agitations one might wish in order to process, in this case, 16 or more grids simultaneously with multiple protocols and identical process timing. So this complicated type of processing can be used for other applications. And Melania McLean at the Stowers Institute published this also last year in Microscopy and Microanalysis in the proceedings. And what was done here was to create a template to process tissue blocks in multiple different ways, and in fact 16 different ways, because this particular tissue there was no good suitable protocol. So this was a way of doing protocol optimization. So here we see 4 tissues by 4 tissues, or 16 different samples of the same tissue that were prepared in 16 different ways. And in this case, it was a planarium worm. So different fixation stains, buffers, and embedding resins were used in this matrix of 16 different ones that were rapidly done. We can also apply this into sort of the materials world and look at some hydrogel formulations, in this case hydrogels that were being investigated by a pharmaceutical company. So these hydrogel particles, these polymer particles, are about 1/2 millimeter in size, and many of them were simply loaded into the capsules, and it was of interest to look at these in the dry state for this particular study. So they were loaded into the capsules, and they were simply directly embedded, actually after an acetone rinse, directly embedded in epoxy. Once they were fully embedded and cured, the capsule could be directly chucked into the microtome, and a block could be trimmed directly through these particles. And here we see many of these hydrogel particles here, which are about 1/2 millimeter or so inside. And we could section through them because for the analyses that were done, FTIR light microscopy and elemental mapping with EDS-SEM, it was important to look at sections because one can get better chemical information and elemental information looking through the section. So here we see some examples of the research results that were obtained here. So we can also do complicated protocols with nanoparticles, in this case a nanoparticle drug formulation. So there were 2 separate drug formulations that were to be analyzed, and they were to be analyzed with 4 different staining protocols. We wanted to prepare them all at once. We wanted to have at least 4 grids per condition, because even with mPrep capsules, you might not have the data that you're looking for, the number of particles on a given grid, or you might potentially drop a grid or so and just to simply have a larger sample size. So in order to prepare 4 grids, 2 grids were prepared in one mPrep G capsule, and 2 grids were inserted into a second mPrep G capsule, and these were then stacked onto a pipetter, again using the mPrep F coupler, and these particle suspensions, the nanoparticle suspensions, were pipetted into the capsule for processing. Now this was done on an 8-channel pipetter, and you can see the one and then the second labeled capsule sitting here over an ordinary 96 well plate with the reagents laid out with a protocol that we can see in a little bit more detail here. So this was the protocol. We have 2 particle types. So half the capsule saw one particle type, half the capsule saw another particle type. These were then adsorbed, and then non-adherent ones were rinsed out by water in all of the capsules. And then we were investigating a positive stain for a particular feature in these nanoparticles. And so half the capsule saw the positive stain and half the capsule saw water. Then another water rinse on all of them to remove the stain. And then half the capsule saw a urinal acetate negative stain, half the capsule saw water, and then a water rinse to remove this. And by doing so, we were able to prepare all 8 preparations simultaneously with 4 grids each in a very simple protocol that made this a lot easier. So let me summarize. Using mPrep capsules, which are purpose-built for microscopy, we can do most any protocol or chemistry. We can do life science sample preparation. I showed tissues, macromolecules. One can do cells. I didn't show that in this particular presentation. We can do materials applications. I showed some nanoparticle applications and a microparticle application, one that was polymeric. One can also do films as well, and they can be oriented for cross-sectional mounting. Using mPrep capsules, we can save labor, time, and expensive reagents. We can make many fewer mistakes where one drops or mixes up grids. We can increase the efficiency for processing a single sample or dozens of samples or even potentially hundreds of samples. It's easy to scale the process from 1s to hundreds of samples. And because we can cut reagent consumption, and I demonstrated dropping consumption by about an order of magnitude, we can be much greener in our laboratory practices by eliminating a lot of the consumption of things like urinal acetate and osmium. They're very easy to use. We can leverage existing labware and tools. There's very little extra materials to buy because many labs have 96 well plates and lab stands, etc. The protocols that we can employ, we can adapt to do things like critical point drying. I showed a cryogenic process. We can immerse the capsules using holders and things. We can transfer to microwaves. And in fact, we can even adapt using the mPrep capsules to run entire experiments in the capsule. And for example, the study that I showed of the negative staining of DNA REC-A complex was to some extent an experiment in a capsule because the adsorption was done in there. And the same with the nanoparticle example where we were able to do some treatment of the specimen within the capsule. And one could extend this into more complicated experiments. And finally, because things are always documented and labeled, this makes it very easy to integrate mPrep processing into laboratory information management systems. If you're a clinical lab, into electronic medical records. If you're a regulated lab and you need to show good laboratory practices or meet CLIA or CAP standards, these can be incorporated there as well. So I certainly thank you for your attention. The webinar will remain open to address any further questions. If you're viewing this via recording, do not hesitate to contact me personally for technical scientific questions, either directly through my email, through our website, And of course, we also have a sales channel that you can reach through our website or directly via email. So I thank you very much for your question, and I will sign off now.